I'm going to be showing you how to do a mini prep using a 24 well block. This is a kind of a medium throughput technique so that you can mini prep a lot of samples at one time. Here we have 14 samples. We have 6, 7, 8, and then there's another row here with another 4. So you can kind of see how many samples there are. If we were to do this all in tubes individually like you've seen before, it would take forever. This is just a little bit more efficient. So first thing we're going to do is we're going to spin it down. I'm going to tear my scale. I'm going to grab one of these little racks. I'm going to put my culture block on the rack and then weigh it. So that's 212.0, and now we're going to have to make our balance. So get another rack, put another 24 well block on it, put it on there, and now we're going to shoot for 212.0. So we just take some water and fill it up until we hit 212. You want to try to fill it up evenly, so I try to go around the edge and then maybe on the inside. I always try to weigh it as evenly as possible because I have this idea that if I don't weigh it evenly, then I may mess up the rotor, the density may have something to do with it. So almost there, and that's probably close enough. So 212.0. Then we just put it into the centrifuge. We're going to close this. We're going to check the rotor, S5700. We're going to put our RPM at 5400, and we're going to spin for 3 minutes. Press enter to lock everything in, hit start. We're going to come back in 3 minutes and we're going to see that our block, instead of having a bunch of culture, is going to have a pellet at the bottom and a bunch of supernatant, and then we'll proceed with the mini prep.
Make sure the rotor is in.
When you hit start, we're going to be watching the speed slowly ramp up. I usually just sit here for a few seconds, keep my hand on the centrifuge, and that will tell me, I can get a sense for the vibration. And you're going to be able to also listen, and if it's off balance, you're really going to start to feel it shake and you're going to start to hear it. So it's going up to speed now, it's hit full speed, so we can be safe, we can be sure that it's safe. Also, you're going to hear that thing that just clicked on, that's the refrigerator, so don't worry about that. Now, if you do have a balance, if you have a really bad balance, this imbalance light is going to light up, it's going to turn red, and it'll just stop. But if you have a kind of a medium imbalance, it will not trigger the safety system, so you're probably going to want to hit stop on your own, which is right there. And if something really bad happens, you might want to hit fast stop. I've never hit that one before, but that'll slow it down a lot faster. So treat this guy with respect. It's a pretty big centrifuge. There's a lot of energy in there. If something goes wrong, it can go terribly wrong. You can look up online. They have pictures of accidents that happen in other labs, and they can really tear up the lab. So we'll be back in a couple minutes to finish up our mini prep.